The overall goal of this experiment is to visualize and quantify endoplasmic reticulum mitochondria interactions using an in situ proximity ligation assay in fixed cells.
This method can help to answer key questions in the biomedical field to better analyze ER mitochondria interactions in fixed cells using a simple immunofluorescence technique, hopefully suggesting new treatment strategies. The main advantage of this technique is that we can visualize and quantify ER mitochondria interactions in fixed cells, and this technique can be applied to paraffin-embedded tissue biopsies of patients.
Using the in situ proximity ligation assay demonstrated in this video, a mouse primary antibody directed against VDAC1 at the outer mitochondrial membrane, and a rabbit primary antibody directed against IP3R1 at the endoplasmic reticulum membrane can bind to their epitopes in proximity at the mitochondrial-associated membrane interface. Proximity ligation probes that have DNA end tails attached and that are directed against mouse and rabbit IgG are added and can form templates for the ligation of connector oligos, and can be amplified and visualized using Texas red oligonucleotides. This can occur only if the two targeted proteins are at a distance below 40 nanometers, in agreement with the distance between both organelle membranes at mitochondrial-associated membrane interface. To fix and block cells for the in situ proximity ligation assay, plate human H7 cells at 150,000 cells per uncoated 35mm glass bottom dish.